So brain-computer interface design and implementation is the topic for today's lecture, where you can see the entire BCI system here, where other things we have already covered a number of times. How do we acquire the stimulus? How do we acquire the signal, and signal goes to computer? Analysis is made. So what kind of hardware is used, how the signal is digitized, what kind of sensors are there, what kind of electrodes are used, how those electrodes are deployed, what are the technologies, how do we capture the signal, if the signal strength is very poor received from the brain, how do we enhance the signal strength, how do we digitize the signal from continuous signal, that is very weak electrical signal, into digital signal, which is interpreted by the computer, so that computer can either generate. In fact, you can see this is an interesting picture. Computer is generating two type of things. You look at either control commands, what we have been talking about for last few lectures. It generates commands with which or by which we can either operate the wheelchair or the robotic arms or prosthetic arms, or we can even use it for NLP kind of applications. So assistive technologies receive the commands, but we are interested today about the design and interface. And of course, the software and hardware part of the BCI is the focus of today's lecture. And the other area, since the picture is included here, other area, as I have been or we have been discussing for last 4 or 5 lectures, you understand very well. Another major task of BCI is to generate some stimulus. The stimulus will be always in the form of visual, signal, or auditory signal, or tactile signal, right? That is, tactile is something which is related with the touch and other stimulus, like auditory related to sound, visual is related to some impression or the picture. So this is how we try to give the feedback. Anyways, so feedback can be given from two ways. One, from stimulus and stimulus generation and the analysis. It can go to the brain signal as a feedback, and from here also it can go back to the signal. But our focus, as I mentioned, is this one. So let's begin with the design and hardware. So what do we understand by BCI system? BCI systems are, you all know, either invasive system or non-invasive system. Most of the systems are based on invasive techniques, where the recording is done in the invasive manner, where what we do that implantable electrodes or grid recordings from the cortex, either as a ECOG, electrocorticogram, and local field potentials. This is a very popular term, local field potential, called as LFPs. Multi-unit activity, this is indicated by MUA, or single unit activity called as SUV. So we get the recordings in the form of these things in all the BCI systems. In non-invasive, this was about invasive. I actually, I just want to correct. Invasive is not popular. Non-invasive is more popular. Most of the BCI systems are based on non-invasive, such as EEG, MEG, functional MRI. EEG is even most popular among all these modalities. And there is one more called as near infrared spectroscopy. So all these background we have though already covered, but since we are talking about design part, we are again covering this. So there are different types of EEG based BCI systems that help in promoting communication or environmental control and rehabilitation, depending on what are the modalities we are using. Modalities means what type of signals we are using. Accordingly, the EEG can generate a command to communicate something, either to control something or for the rehabilitation purpose. Accordingly, there may be different types of systems, there may be different types of designs. For example, there is a term called as SCP. What is SCP? Is selective cerebral perfusion. Selective peripheral perfusion. Next one is SMR, sensory motor rhythm. If you remember, this came with when we were discussing about independent and dependent BCI. So SMR is sensory motor rhythms. P300 is event related potential BCI. P300 is a popular name. We have understood in the last lecture why do we call it as P300 or P200. And then another one is steady state visually or visual evoked potential called as SSVEPs. So these are the examples of different systems utilizing different modalities. And of course, there will be some difference in the design as well. So if you look at the P300, BCI system, where P300 system is, I have told you that what is P300, that the peak is obtained after 300 millisecond, and it is always relative. It may be from either from positive to negative or negative to positive, but there will be change after 300 millisecond. And whenever there is a mental task or any task where the brain is engaged, the task is assigned to the subject, the event related potential will be generated after certain amount of time. That time 300 millisecond is latency here. So stimulus means, say the person is asked here to read some letters or to recognize some letters from the board. So this is also a task where the task may generate some potential called as event related potential. And then other thing is similar, that the data goes to AD converter, then amplifier. You know it very well. It goes to AD converter, analog to digital. Rather, it goes to amplifier, then it goes to analog to digital converter, because the signal strength is weak. So today we are going to talk about this particular area, and the electrode area, which are actually the design and implementation part of BCI. So 300, widely known as P300, which is lead positive component. Why it is lead term? Because after 300 milliseconds, so it is lead positive component evoked in response to an external task relevant stimulus. So the task will be always stimulus external. And when someone is engaged in the task, a stimulus will be, an impression will be generated equivalent to that particular potential. So P300 component is used in order to control. This is actually very popular. You can find a number of research papers and articles on these. P300 is used in order to control devices, even those who are having some disabilities in a smart home or a smart environment system. You can use P300 to either control the devices such as wheelchair. Wheelchairs can be controlled, and P300 can also operate on real and virtual environments and allow the user to use paint interfaces or browse the internet. Even the user can, do some pencil or painting work, and also user can be guided to browse the internet and to do something to do some task. And these kind of control activities are facilitated by the system called as very popularly known as P300 component, or P300 popularly known as P300 BCI. So this is one example of the response, like the task. If you look at here, that if the person is doing nothing, there is no movement, there is no walking, so person is standing. So in that case, you see the response in the different area. What I can say is evoked potential, evoked response potential, right? And look at here, this is the term called as event related potential or evoked potential also. And when say the person is walking on treadmill, and there is no motion but walking, so and for a certain duration, so that particular also creates certain different signal. And if person is moving as well as walking, then this is what is the potential, and the other one is it is walking halfway, and then there is a movement also. So what I mean to say that there are different activities, different types of tasks related to motory tasks. Mainly here, all the tasks are motory tasks because you are walking, you are moving. So it's all motory tasks using your legs. So and therefore you look at here uh, in a particular time interval only, the potential is being generated. This is very interesting because this diagram is for P300. So what is interesting in P300 is you have yesterday in last lecture seen that generally P300 is 300 millisecond, but 300 unlike 200 P200 where the value is exactly 200 millisecond as a latency. But in P300, latency is right from 250 to some 600 or below 600 millisecond. So look at here, the timing which is 276 to 440 millisecond. This is the duration where a peak is coming. And actually, this is a very correct diagram. Look at here that this positive peak is coming. So this is actually the response of the signal generation from the PCI P300 system. So what are the necessary hardware? What are the different types of electrodes which are used? And then how do we capture? How do we sense? How do we detect? How do we handle with noise? How do we amplify? How do we convert? How do we, condition the signal? Are the factors, are the points of today's discussion. So the design theory is a neural interface component which we use in any BCI that gives or that provides an interface between a brain and computer is definitely by using a hardware device so that we can detect brain signal, and the brain signal detected can be sent to other components which are used for analysis and translation to suitable or useful commands. So the interface is connecting brain and so much of hardware, so many hardware components. So those hardware components are part of design theory, right? The discussion is the part of design theory. Different BCI systems will record different types of brain signals from different areas. If you remember in one of our slides, we have seen that we can use a cap. In cap, there will be a number of electrodes, but it is not necessary to capture the signals from all those electrodes. So we can use suitable electrodes from a particular area. So different resolutions also. So there are different brain signals, there are different areas, and there are different resolutions. And we can use them to control different types of effectors. Maybe we can use the command to run the computer cursor or to control the switch or to control the robotic arm. Therefore, the BCI designs and functional requirements, the designs of different BCI systems and their requirements, the functional requirements will not be constant, will not be fixed. Why? Because of different brain signals, because of the area, the number of channels for different modalities, and that to different resolutions. Because of these, the designs are not fixed, that vary over a broad range, and they are actually controlled or driven by BCI's intended use. We also, the BCI design is also, it also depends on what is the user's intention. What does user want to do with the BCI or from the BCI? And therefore intended target population, people in severe disabilities, what are the things, what are the applications, and accordingly design also varies. So BCI system that provides cursor control, for example, for the different people who are having some lateral sclerosis that have a much different design. Say the problem is having some problem in the movement of the arm. The design will be very different from the functional requirements of a person, who is having difficulty to control the arm and hand. So I think you can very well understand this like this. Like this is one example where amyotrophic lateral sclerosis, ALS, where you can find a muscle when it is contracted, and when a person is not able to contract the muscle, the contraction is not possible. In both the cases, the normal nerve cell is like this, and this is not the normal nerve cell where there has been contraction, and this is what happens in the sclerosis patients. So in such study or analysis, the BCI may not be same for what has been highlighted in the depicted in the other picture. In other picture, you can find that, there is a arm where even you can control these joints, a joint 5, 4, 3, 2, and so on. And there will be different types of motors. All those motors at different points can be controlled by different commands, and that too in adaptive manner. So therefore, the design varies. So what is the major challenge in terms of requirement is to develop an effective and versatile interface, because see, robustness is always a challenge in all such task. Versatility, we need to develop an effective, very effective and versatile interface so that we can detect the neural signal, that is brain signal, and that signal can go to signal transduction, means it can be translated, and suitable requirements of BCI can be made. So what is important is how can we design a versatile interface so that whatever is the type of signal, whatever area is covered, whatever number of channels is involved, it can meet the functional requirement, and it can be used to meet the objectives or the intended application of the user. So to solve all these difficulties, because getting versatile BCI to deal with and to address all the different combinations, it's very difficult. So to deal with several primary requirements are needed, like interface must be safe. Whatever versatile interface is being developed or designed must be safe. Neural signals must have sufficient information to support BCI use, and it must be reliable as well. These are the properties. EEG scalp electrode arrays. Now, what do we do in the recording of EEG? We use a cap, and that cap has a number of electrodes. So what do we say? Electrode arrays. So they attach non-invasively to skin to record. What do we record? The field potentials with relatively low information content. I think I do not need to explain again why the low information content comes from very large and widely distributed set of neurons and synapses, because there is a enough gap between the electrode and the neuronal activity inside the brain, because the interface is not direct. The technique is non-invasive. Therefore, the signal is relatively weak, as well as the content is not very strong. We have already discussed about this. Another, the modality we have already covered comes under a semi-invasive, if you remember, electrocardiography, where electrode arrays are surgically positioned, surgically placed on the brain surface to record field potentials, electromagnetic field potentials with moderate information. The information is low in case of non-invasive. In semi-invasive, the information is moderate. Look at here, from smaller, more localized sets of neurons and synapses, right? So you are going to be, little more localized, where in the case of non-invasive, localization is less. It is slightly more in case of semi-invasive, called as ECOG. But when you go for the other one, like microelectrode arrays, these are nothing but miniaturized sensors, miniaturized, acquiring arrays, the data acquiring arrays. You can say microelectrode arrays means a kind of integrated circuit in the form of IC. Miniaturized means, very small sensor, like miniaturized sensor, smart sensor. So miniaturized microelectrode arrays are surgically inserted to cerebral cortex to record neuronal action potentials, which we also called as spikes, and the individual neurons or the local field potentials from from a small, highly localized sets of neurons and synapses that produce high information content. So therefore, accordingly, it's very difficult to have a same design or a common design. I hope what we have covered in this slide is, again, I repeat, for EEG, the area covered is large, the information is low. For ECOG, the area covered becomes more localized, information becomes moderate, little more. And in case of microelectrode arrays, where we directly put, insert the ICs or the arrays inside with the help of surgery, there we get the maximum amount of information, and that becomes highly localized, rather maximally localized in the brain area. Like this is one example where microelectrode array, look at the microelectrode array, which is, being inserted here, and this will be connected to another thing. You can see this is nothing but the electrode. So this electrode will be, as a common electrode, what we use in EEG, but this electrode will be then finally connected with this microelectrode array. So the microelectrode array, look at here, this is going to be inserted, and all these areas will be covering a very, focused area, specific area. So the information which will come will be maximum amount of information in case of microelectrode array. So intracortical BCI neural interfaces will be microelectrode arrays, which can be implanted in the movement related areas of cerebral cortex to record from different individual neurons or local population or a small group of neurons. A larger group of neurons is covered in EEG. The array is designed to penetrate into the cerebral cortex. There are a number of areas. The interesting area is cerebral cortex, where this is penetrated and placed in close proximity to the targeted neuronal potential. So wherever we insert from the penetration takes place towards cerebral cortex. But where do we place the array? The placement of array is made very near to where is our target, which area we are interested to study. If we are interested to study about the the cognitive ability, if we are interested to study about the memory capability, if you are interested to study about working memory, or if you are interested to understand about some learning ability, what is our target, what is our intended target, accordingly we can place. So array is used as any of several types of implantable multi-site recording microelectrodes, that also includes microwire arrays. So microelectrode array, now we are already talking about, we have already started talking about design. So you can see that array is big, it's an essential part of the BCI design, which is used to detect the neuronal signals, and those signals are detected and sent to other components of BCI for processing. So other components are outside the brain, because inside the brain, microelectrode array is there. It brings the signal from what kind of signal? Neuronal activities, brain activities in the form of different modality. Obviously, this is not going to be EEG because we are putting inside the brain called as microelectrode. So here the neuronal activity is not EEG signal. It will be very, very localized field potential that gets captured with the help of microelectrode. So this is the intracortical BCI signal transduction component. Transduction component is nothing but it will actually equivalently convert the signal into the commands. So to create intracortical BCI interface, we are talking about neuronal interface. We started today's discussion with the neuronal interface. So what kind of neuronal interface we want? Complete intracortical BCI neuronal interface. The arrays are integrated with highly specialized low power electronics for signal conditioning. Remember, when we capture the brain signal, even if you capture the ECG or other kind of MRI signal, So when you get the signal from brain or when you get the signal from other parts of the body, it cannot be directly analyzed. It is subjected to a number of signal conditioning and signal processing electronic circuits. So therefore it goes to low power electronics for signal conditioning so that we can make the signal proper. We can remove the noise. Then it goes to the electronic communication circuit, flexible multi-channel cables. Through cables, it is transmitted to connect the microelectrode and electronics. What are used? They are nothing but multi-channel cables. It's not a single channel, but it's a multi-channel so that we can get the information from a number of channels or a number of electrodes associated with hermetic packaging to protect. Why this kind of packaging is needed? You provide the protection to all those channels so that there is no loss of signal which is coming from the brain. So electrodes and which are electronics which are interconnecting the neuronal interface are provided the protection by packaging so that some shielding takes place. The interface can ultimately record the neural signal with very high information content, information which is required for controlling the high dimensionality. Say I was talking about prosthetic limbs. So this kind of development, prosthetic arm, has been done in one of our labs in our institute by another faculty. So prosthetic limbs are associated effectors, and even to control that prosthetic limb, we can generate the commands from that particular signal with the help of the suitable computing algorithm. Microelectrodes. So we are continuing with the microelectrodes, and the main technical requirement for intracortical. Cortical is your ECOG. When we say intracortical means we are going deeper inside. Therefore, because we entered through cortex, cerebral cortex. So therefore we call it, it's not inter, it's a intracortical. So we go deeper to cortical area. So we are therefore calling it as intracortical. So the main technical requirement for this interface is ability to record neuronal spikes or neuronal activities and local field potentials, or either of two. Either we want spikes or local field potentials or both from the target neural population. I have already mentioned about we insert the array through cortex, but it can be placed anywhere where our target is. So neuronal population from which we get the signal in the form of local field potential or spike with sufficient quality. This is very interesting. With a very good quality, adequate quality, stability and longevity. This is another interesting thing. When we get the signal in the form of spike, once it reaches to the surface, once it reaches to the cable and the amplifier stage, if it doesn't sustain, means the longevity is very poor, then there is no use of that local field potential. So all these are taken care of while designing the BCI. The interfaces are very significant because they are used for acquiring the high information content signals which are required for applications such as when we are interested to control robotic arm with, and that too, multiple degrees of freedom. The same robotic arm can be moved with a huge number of degrees of freedom in any orientation, in any direction. The robotic arm can be moved, and therefore you need high amount of information. So high amount of information comes from directly from this kind of interface. So this is one, historical background. In 1950s or in 58 implemented several 80 micrometer stainless. Actually, some work started in 1950s, and Strumwasser in 1958 implanted several 80 micrometer stainless wires into reticular formation of squirrels. The testing was done with the squirrels, and the spike was recorded for 4 days. And what was the steel wire was 80 micrometer wire, and the attempt was made. So long back, this kind of work started. If you remember, we were talking about the historical background of BCI. There also the work started in 50s, and the test was done from animals only. So the approach was then improved by etching a smaller diameter 30 micrometer platinum wires, and which has very narrow tip, thin nip, enabling single unit recording rather than multi-unit for over 3 days. And the functional implantation was done for 3 months, and this was done by Buns et al. In 1973. The first successful chronic microelectrode arrays, which consist of 5 insulated tungsten microwires. They were edged to sharpened tips bundled in plastic tubing. This work was done in 1967. So these are some pictures. Like first one is, you can see 10 mm cable is there, single EEG electrode. The first diagram is nothing but it is just highlighting one electrode. You might have seen, this particular electrode when the doctor uses the stethoscope also to record our, the breathing and all that. When generally doctors use stethoscope, where that electrode they use. So this is the electrode, a single electrode used for the EEG, and it is connected with a cable. I'm sorry. Diagram B that depicts X-ray. X-ray diagram is actually not the actual diagram. It's actually a radiographic diagram of implanted subdual EdTech ECOG, because we slightly go inside. Therefore, it's the X-ray version of image. So electrode grids are there rather than a single electrode. There are grids you look at here, and it is also covering a specific area, as we have mentioned in the previous slide, that some specific localized area is covered, and so it has 4 electrodes and so on. And then C is actually microelectrode array. The development was in 1997. So 10 mm, then 2 cm. Look at 2 cm is the complete area, and this is 2 mm. Now 2 mm is a very small area that is covered by microelectrode arrays. And the other configuration of Michigan microelectrode array, the next one was also microelectrode array. You can see is in the form of chip only, IC only, that goes inside the cortex, cerebral cortex. So these are the different, the electrodes which are used in capturing the, the signal. Those who are doing with some electronics, they can even try, of doing something, designing some hardware, implementing some smart sensor, miniaturized sensor, so that the weak signal can be captured. And of course, you cannot try with this because you don't have access to, or you are not allowed to do surgery. But of course, one can start with even the person who is interested to work on, some hardware. They can think of, development of developing this kind of electrode where a very weak signal can be recorded and the captured. So what are the characteristics of those arrays? The arrays are generally characterized in terms of what are the materials with which arrays are fabricated, and what are the technologies of fabrication, what are the packaging methods and the component, how the other components, all the components of the arrays are integrated together. Accordingly, this is how you can characterize the arrays, how good the array is, how better is the array, how well the array is working, depending on all these characteristics. So while designing the BCI, what is your intended use? Accordingly, the array has to be characterized. Array has to be chosen based on its characteristics or the parameters. So array is also categorized by whether it is designed or fabricated using bundle of microwires or through microelectrode mechanical systems. I hope all those who are having some background from electronics or electrical, they all know about the MEMS very well. In fact, I always tell that in your smartphone, you have, 20 or more than 20 smart sensors are there. So those sensors are actually in the form of MEMS technology, micro electromechanical systems. Otherwise, how those ICs or how those sensors, more than 20 to 25 sensors can be fabricated into a small device called as your smartphone. So these are the technologies, and accordingly, the arrays are of different types. Microfabrication processes are a broad class of technologies that involve microfabrication of silicon and polymer structures for, I was telling, miniaturized sensors, actuators, and integrated micro subsystems. So the MEMS microelectrode arrays, those which are based on MEMS technology, are micro machined wafer. I was telling you it's nothing but wafer means very thin IC, integrated circuit kind of thing from silicon and or polymer substrates, whereas micro wire arrays are assembled from individual micro wires. I think here one question I can ask you, polymer is okay, but when you talk about semiconductor, there are silicon and germanium, right? So you can either choose silicon as well as polymer substrate, which is actually, a kind of compound material, compound semiconductor material, or either of these two. But we generally, do not use the germanium, though the germanium is also semiconductor material. Would you like to answer why only silicon is used in the wafer level? Why not germanium? I can pause for 10 seconds. Would you like to answer why not germanium? Any one of you, please. I would be happy to at least listen to your voice. Anyway, during the break, you can think of answering. Okay, germanium is generally not preferred, and silicon is always preferred at wafer level. It is mainly because of one factor, which is called as stability of silicon is very, very high as compared to germanium. The stability against variation in temperature and against other variation in other factors, the variation is very less in silicon as compared to germanium. Though germanium has higher conductivity, hope you are aware. Germanium has higher conductivity, less threshold potential, but we use silicon in the wafer. Anyway, let's not go into the electronics more details. So microelectrode array, what we are discussing is the characteristics. Characteristics are very important while choosing a design or while designing the BCI. It can be also categorized by what is the fabrication process, like planar process, thin film technology, or micro machine MEMS technology, and what are the materials, like whether we are using platinum or iridium electrode sites, or polymer or inorganic dielectric materials, and also depending on assembly, how do we assemble, how are we packaging accordingly. These are, characterized like. I have included two diagrams just to tell you that look at the second one where there is a MEMS dry electrode. There are different types of electrodes. Now there are wireless devices, wireless, MEMS can also be used. There is no need of, in fact, bringing through the cable. If you are, I can tell you, those who are very interested to study and know about BCI and where is the use of BCI. A lot of studies going on, the drivers who are driving for a long time, if they have, what I can say, if they are feeling sleepy and they go into sleep, how alert can be generated? Can we read their brain, and then can we understand that some abnormal thing is happening? His attention level is decaying, and we can generate the alarm. So in that case, you do not need to connect some cables. You can just, with the help of either Bluetooth or some Wi-Fi technology, wireless or some other technology, you can just get the signal and subject to other device called as the device may be a digital signal processor that may have all the three things. It definitely will have some battery operated device. It may have amplifier. It may have analog to digital converter, and it may also have that wireless protocol. So this kind of application is also done, but it can be also brought with the help of wired technology as well as the wireless technology. Similarly, here also you can see that Here the dry electrodes are placed. You are getting the signal, and 65 by 40 is the dimension of same thing. And what is interesting here is this is nothing but some part of the second diagram only. You can see the cap. Inside the cap only the device can be what is that device? This device, remember, Bluetooth device can be kept inside the cap so that the person who is driving is wearing this cap, his signal is recorded, and we can again get this signal which is being analyzed or being captured in this Bluetooth device to either our cell phone or some computing device. So through cell phone, we can get the simulated signal. We can understand that his attention level is changing, and based on that BCI interface, some alert can be generated by the mobile phone, or through his cell phone, some alarm can be generated, and this person can be brought into some normalcy so that the accident could be averted, could be avoided like this. So let's not talk much about all the processes we have been talking about. What is important is the technology. What is important is the design of these devices. So this is one kind of design which is implemented wirelessly without any wire. The first level differentiates between bundles of microwires and technologies. This is one diagram actually. It is talking about the first level is between the microwires and the bundle of wires or MEMS technology. So it is implemented in two ways: implantable arrays which can be implanted inside the brain are of two types, either MEMS or bundle of wires. And then they are further divided. Look at surface micro machine, bulk micro machine, like bundle of wires, microwire array, neurotropic array, and others. And similarly, these surface micro machined MEMS technology arrays are of some silicon substrate, where substrate is generally the base material, wafer material on which your design is based. It may be polymer, it may be ceramic, and other similar materials which are used. Now, after discussing about arrays, let's talk about a signal registration. The biopotential at the electrode surface, which we record with the help of electrode, that is actually the combined outcome of neuronal activities, the neuronal sources, and whatever activities are there in the vicinity of the electrode, they are conducted through the encompassing brain tissue. It comes through brain tissue, through the conducting path, and whatever points are there in the vicinity, they are connected. And from those points, it finally comes to the surface of the electrode. The brain tissue is a volume conductor. Why the brain tissue is being talked about? Through brain tissue, it comes the signal to the surface of the electrode. So it acts as a conductor. Why do we need conductor? Because we are getting all, conducting signal, electrical signal or electromagnetic signal. So we need conducting path, and the conducting path is provided by the brain tissues. It is assumed to be that path can be linear, resistive, homogeneous, but not necessarily isotropic. This is the property of tissues. For implantable electrodes, bipotential characteristics which are recorded may be either spike amplitude or waveforms or LFPs, what is called as local field potential. So rather than getting local field potential, we can also get local field potential spectra. Spectra means over a wide range of frequency of the signal, what is the variation of local field potential. So rather than getting single peak, double peak, or three peaks, we can get the spectra, and which is affected, of course, by the reactive tissue responses and develop over the time that change the local tissue conductivity. The biopotential of microelectrode site is transduced to the electronic current. This is the phenomenon, this is a process, very simple. The conduction take place and electrode traced by complex but electrochemical processes at the electrode tissue interface, and it comes through then extracellular X-ray potentials. They are having amplitude from 50 to 500 microvolt, and the bandwidth of this one is talking about those potentials, extracellular potential. LFPs are useful for BCI having amplitudes 10 to 800 microvolt, and the bandwidth may be from 0.1 to 200 hertz of all those local field potentials. Not talking about since the design also depends on the type of electrode, the type of interface, but the same time in the beginning we have discussed design also depends on the modality, the type of signal. So let us talk very briefly about EEG versus MEG, which is better and which is and why EEG is popular. So MEG is magnetoencephalograph. The less amount of research can be found in this particular modality. And the MEG signals are obtained by dipole current sources. Correct. Dipole, you know about the magnetic field. There are always two dipoles where polarization happens. And then, the polarization and the loop actually goes from North Pole to South Pole. So there is always a dipole, and that is produced by neuronal activities and the principles, the fundamentals which determine the distribution through the head or the dipole current fundamental. EEG is very inexpensive, is very convenient and amenable to the diverse environments, and it is now coming portable as well and has been developed commercially into portable and even wireless designs for a number of purposes and the applications unlike MEG. In case of EEG, there is no dipole. And there are research groups which are actively working on MEG based research, MEG based BCI, and MEG instrumentation is expensive and cumbersome and not practical for everyday use. It's because of the magnetism involved. As a result, MEG is used mainly as of now, as a research tool for BCI technology is being studied and being used, not for the commercial implementation. So this is one picture, interesting picture that is depicting one kind of a difference between MEG and EEG. You look at EEG where the ions are very important at different surfaces. So whenever you understand there are ions, then it develops some electric potential. But here there are, look at the magnetic effect. If you have seen the response or the dipole response or polarization response of a magnet. So these are kind of that field patterns which are generated in the in case of MEG, whereas the fundamental principle behind generating the electrical potential is highlighted in the second picture. EEG recording then comes to EEG recording. So look at this interesting diagram. We are talking about the hardware design. We are talking about design of BCI. So look at here. What is interesting you can find is, I think in coming slides, we will discuss a little more about this. So if you can find that even if you are recording a signal, there are three, rather electrodes. Look at here. This is one electrode. This is another electrode, and third electrode is always a ground electrode. Remember, this is a bipolar recording. There may be unipolar as well. We will talk about unipolar and bipolar recording, but in this case, what has been depicted in the picture is bipolar recording. So in bipolar, what happens? There are two electrodes. There will be always a reference electrode, generally a ground electrode. So these bipolar electrodes will read certain potential. For example, the differential amplifier. I hope all those who are having some electronics background, they understand differential amplifier. One of the popular amplifier is, if you can, I wanted to connect iPad today, but it is not, much writing is not needed. There is a very popular IC number, you may be aware, 741. IC 741 is, operational amplifier. So operational amplifier is nothing but a differential amplifier where it produces, rather it is the amplified version of the difference between the two input. So say this particular reads 1 millivolt, other one reads minus 1 millivolt. So what will happen? The output will be minus 1 minus minus 1 is 2 millivolt, and it can be further even amplified by the differential amplifier. So what is the necessary hardware here? The first thing is, the electrodes which are placed on the skull or they are implemented on a cap-like structure. Then it goes to differential amplifier where amplification of signal takes place, because we know that these electrodes are very far from the neuronal activities. So the signal strength is weak. We amplify and we also then we can pass through suitable filters. If you can remember, I have already discussed about the artifacts, the noise, eye blinking, or any unwanted effect during the recording of the signal can be filtered. If you want to separate the high frequency content, if you want to eliminate or rather attenuate, you can use high pass filter. If you want to attenuate very low frequency signal, then you can use low pass filter. And then finally, again, after passing through the filter, signal strength may decay, slightly, because it is again an electronic circuit. So you can again pass through the gain certain amplifying device. If it is required, then you can pass through ADC. And then after, getting the signal, making it proper, filtering it, removing unwanted signal, you can then finally convert into digital. You cannot directly convert into digital here because unwanted signal will be also unnecessarily converted. Then you are, spending extra cost on the hardware and your timing will be also more. So the ADC will be converting analog into digital, and then that digital signal goes to the signal processing where you can understand the signal, you can understand the waveforms, you can extract the alpha beta patterns, you can understand different features, you can extract the features, you can then send those features for the classification tasks. So these 3, 4 stages are very important in designing a BCI electrodes, amplifiers, filters, and analog to digital converters. Of course, then it goes to signal processing. So signal processing is a broad area that of course involves feature extraction, feature extraction, and that then is a group together as a feature vectors, and they are sent to the next stage. So each recording involves 3 electrodes. I have already explained: ground electrode and 2 recording electrodes. Ground electrode is connected to the amplifier ground. If you see the IC741, there will be always a ground terminal here. So in all the amplifiers, there will be requirement of grounding. So the ground electrode is connected to the ground of amplifier, which is isolated from the ground of the power supply. And then the potential difference, actually V1 minus V2 or V2 minus V1, is actually obtained from these 2 electrodes, and you can send then biological artifact. I have already explained. If there is any biological artifact, you can pass or you can attenuate through the filters. So environmental electric and magnetic fields can also generate scalp potentials. Remember, when you are capturing or recording the biomedical signal, the in the vicinity, in the environment, the electric and magnetic fields which are present nearby can also impact, so can also generate some scalp potential, mostly due to the capacitive coupling of body and electrode leads to the ambient electromagnetic field that may be related to power lines and other electrical equipment. And therefore it becomes very important to filter your signal and pre-process the signal to make it more accurate. Potential difference at scalp 1 and 2 is, external ground is V1 plus V2 is a common. So what is the potential in the first node? V1 plus V common. V common is coming from the reference voltage. V2 plus V common. So what do we do? That when we, use the differential amplifier, subtract this V2 minus this one. So common voltage is automatically generated. So if you have studied about operational amplifier, it does not amplify the common mode signal. It always amplifies the differential modes. So EEG systems use generally the differential amplifiers, which are actually designed to reject spatially constant the common mode potential and amplify the difference potential, which is between the pair of 2 locations, such as output voltage that is proportional to the scalp potential difference, which is generated within the body. So the ground electrode is placed. So where can we place the ground electrode? Because you are implementing and generating, designing. So it can be placed on a scalp or a nose or a neck or a neck of your body. that can provide reference voltage. So therefore, it may not be zero like what we know about the ground voltage. It may be some common voltage, but it is nullified because of this process. V1 plus VCM plus V2 plus VCM. And the amplifier to prevent amplifier drifts. What is the significance of this? It always helps to avoid amplifier drift and facilitate better common mode rejection. There is an important parameter in operational amplifier called as common mode rejection ratio, indicated by CMRR. Probably the next statement is about, I think is given in the next slide. The ground serves as the reference for differential amplifier. Remaining unwanted contribution from the common mode is mainly due to unequal contact impedances of two recording electrodes. So this is how, in the right hand side, you can understand the cap with built-in electrodes. Look at these electrodes. And if you remember, in the last lecture, we were trying to understand what is F7, what is F3, what is F5. We have understood and giving a specific montage. What is montage is there in next slide. Actually, that's a structure of electrode, arrangement of electrode is called as montage. We will discuss in the next slide. So in the left side, cap with many sides. Look at, there are so many sides for individually screwed in electrodes. There are so many sides, whereas in this case, there is a limited one in the cap. So traditional, generally conventional EEG provides guidelines for contact impedances, because impedance is also very important. That is generally less than 10 kilo ohm. This can be achieved by braiding the scalp at the electrode and using a conductive gel or paste between the electrode and the scalp. So when we use modern amplifiers, they are having large input impedances. The amplifiers like a differential amplifier. If you have studied about operational amplifier, ideal operational amplifier, again, I can allow you to come in. What are the properties of ideal operational amplifier? There are many. Common mode rejection ratio must be infinite. The gain must be infinite. Input impedance must be an infinite. Output impedance must be zero. That is ideal. Practically, input impedance must be very high. So you can see 200 mega ohm and the contact impedance 30 to 50 kilo ohm can be easily tolerated without degrading the EEG quality in the output. So these are electrode, placements. If you can recall from the previous lecture, you could find that these are the two ear positions, ear lobes, and this is the nose area, and this is the just opposite to the nose area, the back side you can see. And these two are your two ear lobes. And accordingly, like parietal lobes, you see here P1, P3, P5, P7, and this is on the center. So 2, 4, 6, 8, which are on the right side, even numbers, look at, they are on the other side, like this side, probably in this earlobe side, and all even look at here odd numbers in this case. Similarly, then other things also you can see the nomenclature. So this is a picture that is connecting the hardware. You can what I was explaining in the previous diagram, it is explained here. There is a ground potential, some potential is grounded, that is FPZ, and there are two electrodes which are, probably FC is frontal cortex and CP is it's parietal and cortex. This is between you see the cortex area and parietal lobe. So between these CP1, CP2, CP3. So you see here CP2, 4, 6, 8 are and 1, 3, 5 are this side. Correct. So the two electrode potentials are coming to the amplifier. There is a ground, goes to filter, ADC. And so finally, what is the output? Look at here, CP3 minus FC3. Why? This is CP3, which is being recorded, and this is FC3. So this difference comes here, and then this amplifier, what we are talking about as one block, this is one sample of, a hardware, the circuit diagram of the biosignal amplifier. Why we are calling biosignal amplifier? It's actually electronic amplifier used to amplify the biosignal. So this is nothing but an arrangement of two, it's nothing but a differential amplifier. That's all. T signals are coming and they are finally going here. So generally what happens, they are going, they're passed through two buffers. Right, these are the two buffers where they are passed through. This is also an operational amplifier where there is direct feedback element and the zero resistance there in the feedback that becomes a buffer amplifier. So both the signals directly go here and then you get the inputs and the difference is amplified and you finally get the output voltage here. So gain is very high, maybe 1,000 times or more than 1,000 times. You can amplify whatever is your, because see, if your signal is in few millivolts, it's very difficult to quantify and ADC can work. So that digital equivalent signal for further analysis and feature extraction becomes very weak. So we need to actually amplify. Buffering stage, you can definitely answer this why buffer is needed because you can never give input directly to the amplifier. So the signal will be stored for some time. Probably the amplifier is working with the previous data. So for that, it will be always waiting here in the buffer stage. So buffers are also used for therefore, impedance matching so that there is no overload in the output. For that, buffer is always the device which is used. So this is a general flow of work, which is not new for us. We have been talking about VD conversion, amplifier, mental task or a protocol or a ERP. So it goes to signal processing, feature extraction. You can also select the feature. If you remember today, I started with this only. So we have a specific work on feature selection and channel selection. So that I'm going to take up in the next lecture. So we suitably select the features. Even here, we can also work on channel selection. So that is also a interesting work. So I'll be talking about channel selection and feature selection in my next lecture with the practical application and examples of using various optimization and ML models. So then this goes to some algorithm where the software part, the mathematical models come into picture, and then this is very well known to you that what we are doing, either generating commands, controlling something, or we are trying to communicate. We can communicate to some other device also, or we can communicate back to the brain as well. After talking about interface, electrode, amplifier, and filters, let's briefly talk about hardware and software because while talking about designing the BCI, the discussion on hardware and software are equally important because that ensures effective functioning of BCI. Without proper hardware interface, without a proper software, or without certain protocol, the functioning won't be appropriate or proper. So powerful, inexpensive hardware will be always required, which is not very expensive so that it can be implemented, the implementation may reach to the portable, level, and it must be powerful software. And these hardware are the important factors for the explosive growth in the BCI research and development. And therefore, you will find that these days you can even find a device. There is one device in one of the research paper where I talked about in my probably second lecture. There are papers which are doing research on learning impact during the online lectures. The device name is Mindset. You can also search that. It's probably $100 USD, 100 USD worth device. So, such kind of hardware, such kind of electrodes, such kind of EEG recorders are coming. It's because of powerful and inexpensive hardware, and that is very needed. And as well as the software interface, you can connect with your computer and laptop and bring the signal and analyze. So hardware provides the physical means. No need to explain why the brain signals, how the brain signals are acquired, how they are digitized, how they are stored, how they're analyzed. For all these things, some hardware will be needed. It also includes a number of sensors, amplifiers, analog to digital converters, computing devices, your laptop, computer, and the processes that digitizing the signal. Cables are also the part of hardware. Software, what is the role of software? You know what is the role of software. So here also it controls the storage and analysis of digitized signals. Finally, your ADC has converted your signal, the brain signal in the form of digital. So it has to be stored, it has to be analyzed, it has to be processed by using suitable software. So the conversion into suitable command as per the intent of a user will be done by the software. So software is nothing but mathematics, a mathematical program, is a mathematical model that helps in converting into suitable commands or communication protocols based on what is the user's intention or the application of the BCI. What are the challenges? It's not yet very clear that which are the sensor modalities or which are the brain signals or which processing methods are more optimal, rather optimal. So this is really, I'm sorry, is really a challenging task that which modality is more appropriate, more optimal, which brain signal is more optimal. Of course, we know that when we go to the invasive manner, we'll get the maximum amount of information, but which is optimal for which kind of application, keeping the risk factor, the risk of surgery all into consideration. What is the optimal? What is the optimal processing method? These are all the challenging questions in the design of BCI. So therefore, it becomes essential to evaluate and compare the efficacy of those signals, those sensors, and even the processing methods. And for that, there are a lot of optimization methods which are used. So you look at here, it becomes important to optimize the processes which transform new BCI designs into functioning implementations. In early computer development, like introduction of RS-232 is a very popular name which is used to, used for a serial connectivity in the computer, RS-232 interface and connectors based on that standard remove the need to develop new communication protocol for every new printer or other device. Similarly, the use of appropriate set of specifications, tools for hardware and software are also very important. Like this is an interesting diagram where you can find this part is hardware, reference electrode, frontal electrode, and the ground. These are all the hardwares, right? And this side you have a signal transmission, then you get data collection analysis. So here there is a role of software. So what is the prototype development? What is stimulus presentation? What will be the analysis? What are those models? How optimization will be used? Which are the signals which are more appropriate for the application? So all these specifications related to all the hardware, the software protocols are all the important considerations, and if you don't consider them very appropriately, it poses the challenge in the analysis of the signal. So specifications of the connectors, implanted devices could be also used for wide range of BCI applications. Connector specification implementation may be physical connectors of different sizes, adapters for common connectors, and so forth. BCI software technical specification may include set of software implementation that realize the technical specifications. Like, these are different types of sensors. You see, all there are different types of sensors which are available. So sensors are basically used to detect physiological signals, which are then converted into voltages and they are amplified, digitized for processing, and then finally they are converted into output signals. So for that, there are a number of variety of biosignal sensors are available, and this is a very difficult thing that which is better, which is optimal. So many of the BCI systems use, most of the systems use electrophysiological sensors, and they are mainly EEG electrodes or ECOG electrodes. They're all electrodes, like if electrophysiological covers everything. Generally, we prefer EEG. So, but they are all EEG, ECOG, and microneedle or wire electrodes. So the last one is always related with microarray. So BCI can also use sensors that detect magnetic or metabolic process. If you remember, we were talking about metabolism where flow of oxygen, it creates the impression in the spectroscopy. So therefore, functional near infrared, functional magnetic resonance imaging, encephalograph, all these are under these modalities where, so there are a lot of, different varieties, different types of sensors which are available. Like, I think this picture already had come in the previous slide, example of single electrode and X-ray of ECOG, microelectrode, and there is a one more configuration of microelectrode having very less small dimension. There are other different types of, these are only the EEG sensors. You look at the first diagram. So this is how the structure, there are different types of designs of cap, which are, which the subjects have to wear. And here you can connect your electrode. You look at here, this is the electrode. You can connect where, whichever area you are interested, you are targeting it, you can do. So if this electrode, which you are seeing, what is the nature of electrode, how it is designed, you look at here. This is a foam substrate, the outer surface. It is nothing but to protect the electrode because that's a conducting element, EEG electrode, and length is approximately this one, diameter is 12 mm. And this is a earpiece where also, from there also, there are a lot of, electrodes which are coming from there. You can get some even the brain related signal with the help of earpiece as well. So you can also insert inside the EEG, and ear EEG sensor can also be obtained. And of course you can. And if you remember, I was telling in the previous slide, the ground electrode can be connected anywhere. It can be connected on nose tip, it can be connected on ear, it can be connected on neck, just to get a reference voltage. So you see here, one electrode is connected here. This is completely non-invasive, and you can find the variation of your signal. Of course, I believe this variation is in millivolt, 20 millivolt to minus 20 millivolt, and this side it is in second, like 10 to the power minus 5, 8, I think probably is 10 to the power 8 into 10 to the power minus 5, something like this, or zero 0.8. So the timing may be second, or timing may be millisecond. So this is how you use different types of EEG sensors. So we can close here. In all electrophysiological techniques, electrodes are used to detect electrical voltages generated by neuronal activities, and then they are passed to amplification. Different electrodes have similar operating principles, function, and their design, size, material, geometry may be all different depending on recording location where you want to apply. Let's say inside the nose, inside the ear, or over the scalp, or cortical surface, or within the cortical, inside the cortex, whatever. EEG recordings, electrodes are placed on a scalp and not in the direct electrical contact. Conductive tissue. So conductive electrical gel I was talking about. This gel is applied between the electrode and the skin so that the conducting path is established and you can get the signal. Dry electrodes do not use gels, and actually they are currently under the development. Of course, there are research on dry electrodes, but until there is an conductive interface, it's very difficult to get. So even the development activities are going on for implementation of dry electrodes. I'm talking about the for brain signal recording. Other sensors such as ECOG, microelectrodes, arrays, they are in direct contact. So conducive tissue or gel, these kind of things, the necessity doesn't arise in this particular case. So systems you will find when you Google 1020 and 1010 system. But 1020 system is very popular, very, very popular. And what is this 1020 system is nothing but, intra-electrode, rather inter-electrode variation will be 10% of your total dimension. Means if this is a total dimension from this end to this end. So between the two electrode, the 10% of total dimension is there. Such thing kind of characteristic is there. I'm also not an expert of designing the 1020 system, but when you get some information about what is 1020 system. So whatever is the dimension, so electrodes are placed at 10 to 20% of, of the maximum dimension. the placement is related to position. Placement position is related to the system anyway. So if you see this particular position, it is already repeating. Look at here, right? But if you can count here, how many electrodes are there? So you have two, and then seven, then 9, 11, and then 14, then 17, then 2 and 19, 21. So there is nothing like no relation with 10, 20. It is related with this one. Here in this case, 2, 4, 7, 9, 12, 14, 17, and 19. So there is no relationship. What is there is and I have already indicated, explained to you that the parietal lobe is this area, occipital, which is vision related, is this area, temporal is this particular area, and frontal is this particular area, isn't it? So, and it is actually divided it has been indicated just like this. This side is your nasal area, this side is the inion area, these two are the earlobe area. Accordingly, different functionalities are there in different parts. So wherever you are interested, wherever your target is, you can use the electrodes in those areas and you can capture the signal. And nomenclature of all these names are related with, I have already told you about the frontal area, parietal area, temporal area, and occipital. Look at here, occipital, then temporal, then parietal area, something like this. You see parietal area. This one, this area is consisting of parietal area. So these are the electrodes, and again you can increase the number of electrodes. And probably what is C, maybe central cortex area. Occipital is O, F is a frontal, P is a parietal, like this. T is temporal, of course. T is also there. T is a temporal area. Yeah. So this side, the green colored area is a temporal area. Look at here, T4, T6, and T3, T5, and all these entire area is a frontal area. Where you have this is on center, and then 1, 3, 7 are the odd numbers this side, and 2, 4, 8 are even number this side. And similarly, the yellow color area is a parietal area, and this light blue color, light pink color is of occipital area. All right, so montages. If you remember, in one of the slide, there was a term like montage. I was telling you what is that? It's nothing but I was telling you arrangement of electrode. Electrode configurations are actually called as montages, and they depend on the configurations depend on electrode type of electrode, location of electrode, kind of information which is obtained from the electrode. This is how the configuration changes. So the most commonly used configurations are, this term also has already appeared in the previous slide, monopolar and bipolar. Generally, maximum recordings of voltages are bipolar, where you use 3 electrodes. Remember, 2 electrodes, because one electrode which is common as a ground electrode, that is common for all. So they measure a potential difference between the 2 locations, means 2 locations of your brain. In bipolar montage is a configuration. The signal recorded from each electrode is the potential difference. So it finally comes to channel cable. What is that cable voltage? The voltage which is coming from the cable, that is actually the potential difference between it and another electrode placed over a different brain area, different part of the brain. In a monopolar montage, what happens? It is not bipolar. The signal recorded from each electrode is the potential difference between it and the common electrode. So remember what happens in the bipolar. We have seen the calculation. The first voltage is the voltage of first electrode plus common voltage. Second is the second voltage plus common voltage. But the final one is the difference. So common voltage is nullified. You get V1 minus V2. In case of monopolar, it is always the potential difference between that electrode from which you are recording and the common electrode, which is placed at some distance from the brain. But most of the recordings are bipolar. So reference electrode is neutral, mostly insensitive to brain activity. So that is generally not affected. And therefore, if you have noted that sometime we can connect that at our nose or ear or neck, because see, neck area is hardly affected by the brain area. Even the tip of the nose is not affected by the, the brain activity. There are studies, I can tell you, where the quality of the sleep and it also is affected by the brain signal, and that is related with your respiration and the breathing. But when you connect your electrode at the tip of your nose, nothing is going to do with the breathing or the respiration signal. So in bipolar montage, every single electrode is referenced to another single electrode, and in monopolar, every single electrode is referenced to a neutral electrode. So with respect to neutral, every electrode is seen, and in the case of bipolar, every electrode is seen with respect to other electrode because neutral gets nullified. This is one example, how do we compute the final voltage? Signal and reference electrodes are often referred as active and inactive electrodes. Like see this one, look at the potential difference. This is exactly the EEG signal looks like. 7 microvolt is a frontal, the fourth electrode of frontal area. Then similarly, this, the central area, 0 microvolts, the difference will be 7 microvolt. 7 plus 7, suppose they are at same potential, you will not get anything, 0. And therefore, you look at here, no any peak or no any variation in the amplitude. Similarly, the third one, which is actually 7 microvolt minus 7 microvolt. So it is actually peak to peak. 7 to minus 7. So the peak to peak is minus 7, 14 microvolt. So it is actually 7 microvolt to next peak is minus 7 microvolt. And this is another interesting diagram, average reference. Sometime the reference is computed as the average of all electrodes. So FP1 minus this one, F3 minus this one, and C3 minus this reference. So this is how the voltages are computed, and they are passed through the cable, and that particular voltage goes to the amplifier. This difference signal goes to the amplifier. So the design of electrode, if you look at this interesting diagram, you see here, this is a skin of electrode, and these are the electrode points which are connected. Sorry, this is the skin of the body, means the brain where biopotential sources, the neuronal activities are here. So what do we want? We want the conductivity from to be recorded from the neural activities. So this is, constituting a volume of conductor where there is in this area, there is some neuronal effect which is recorded. So there is some design of, and then we connect the signal from electrode to front end electronics. This is nothing but the amplifier, ADC, filters. They are all combined called as front end electronics. So EEG electrodes can be made can be made up of different metal or metal salt combinations. The choice of the metal affects the type and quality of the recording. If you want better quality, the material will change. The metal, the type of metal will change. Commonly used EEG electrodes are made up of gold. Of course, you can understand, gold and silver have very high conductivity, so it will capture the signal. So gold is there, tin is there, and you know the price of gold as well. Silver chloride, it is just to provide the conductivity. So you have seen metal salt combination also, correct? So silver chloride, it is not sodium chloride. I'm sorry, it is silver chloride. So because of that salt combination, it is coming as silver chloride. So gold and tin electrodes are maintenance free and have a good frequency response for most of the EEG purpose. Remember, when we use the electrode, what is important is the stable frequency response. Over a range of frequency, the amplitude must be stable. So the frequency response is good. For recording EEG signals that include frequencies below 0.1 Hertz, a very less frequency, silver and silver chloride electrodes provide superior result. You can understand these extremely low frequency, and we know that the silver has very high conductivity, and therefore we use silver kind of materials. So therefore, what is our application? What type of quality of recording we want? Accordingly, the design of electrode. So this is what I was telling you. If you have noticed in the previous slide, that you can think of designing, those who are doing on hardware, you can think of designing a small electrode, and you can test whether it captures some brain activity when you are smiling, when you are crying, when you are laughing, when you are doing something or thinking deeply. then just capture the signal and just get into the oscilloscope. Oscilloscope, but oscilloscope has to measure that much voltage, in terms of milli and microvolts. So you can think of using other devices like digital storage oscilloscope and so on. So the electrodes are of two types: active electrodes and passive electrodes. Passive electrodes are metal disc made up of tin, silver, silver chloride, gold plated silver connected to the amplifier by the cable. Anyway, the electrodes will be always connected to the amplifiers. It goes finally to the amplifiers, and the interface is provided between the skin and the gel through gel, and so that conductivity is provided. We have already covered. The brain signal strength is very small, so and even the strength is very small. Now, why do we use amplifier? Because when we take the small signal, that can be corrupted, contaminated by a movement of the cable or some artifact, and therefore we amplify. So the contamination may be by environmental electromagnetic noise such as power line interference. Therefore, what we do generally is a cable which is connecting the electrode, it is always protected by some material, some protection layer, some insulating layer will be protecting, so that even if there is a interference, environmental change or interference or noise, that cannot cannot contaminate the signal. So the passive electrodes are relatively inexpensive and used in majority of clinical research and EEG recordings. Correct. And this is one example. I think we have already covered active electrode. There is one more elective. This is nothing but one same person and actually indicating two different points. This side one electrode and that side one electrode. Ground electrode is connected here, and this is a reference electrode. So you can get left EEG, you can get right EEG, and this is something which is related with, I think, why it is ECG. ECG is generally electrocardiograph. So these are the two different points. Okay, we are interested in EEG. So active electrodes have preamplifier. The difference between active and passive is in active electrode, there is an inbuilt preamplifier, and that can amplify your signal maybe in the range 1 to 10, built inside the electrode. This makes electrode less sensitive to such factors like noise and cable movement, because there is some inbuilt amplification. So a signal strength which is received by active electrode is little larger as compared to passive case. So the contamination possibility becomes less. Active electrodes therefore perform well in environment with higher environmental noise or higher skin impedance, electrode skin impedance. These electrodes also require gel at the interface between the scalp and the electrode. Gel is serving the different purpose, just to provide interface between the skin and the other part, like hardware. And what is another interesting thing is there is always an interface between skin and electrode. And in order to reduce that impedance, because there will be always an obstruction, impedance between the resistance, impedance will be there between skin and electrode junction, interface, and that impedance is between scalp and the electrode as well. So when you put the gel, actually, it determines the quality of EEG recording. More will be the impedance, poorer will be the quality, because the actual strength of the neuronal activity will not reach if the impedance is more. So conducting electrode gel placed between the electrode and scalp decreases the impedance. Obviously, when you place some conducting gel, it will reduce the impedance, and so that more current will be allowed to travel and easily and smoothly from scalp to the sensor. So to the sensor level, the neural activity reaches easily, reducing the impedance. So the role of gel is to reduce the impedance. A very simple thing. The impedance is measured between two electrodes placed on the scalp. We have already discussed about this. The impedance is frequency dependent. Remember, it is not independent of frequency. It is dependent on frequency. At a different frequency, different just different frequencies, different effect will be there on the impedance. Generally in the range of 20 hertz. It also depends on the surface area of electrode and condition and preparation of the skin, properties of gel, what kind of gel we are putting, amount of time when we are recording, how much time we are placing the electrode on the scalp. All these factors determine the impedance. Electrode gels containing sodium chloride in concentration of 5 to 10% can also help in reducing the impedance. So when we with the conducting gel, we can also mix some sodium chloride 5 to 10%. It also helps in further reducing the impedance. For good EEG recording, the impedance is expected to be below 5 kilo. The impedance between the scalp and the sensor it has to be below 5 kilo ohm. Unstable placement, if your placement is not proper, so that is also common factor of a poor or artifact EEG recording. So there will be more predominance of artifact that will further reduce the quality of your signal. So many research and clinical groups are commercially available electrode caps that allow for fast and accurately position placement. So you just wear a cap. Do not worry about positioning the electrodes very properly and appropriately. Just you wear the cap. So there are some commercially available designs in terms of electrode caps. Just wear that, and wherever you want to cover that signal, that electrodes only you insert in the slots of the given slots of the caps. Most caps use standard international 1020 system I was talking about in the previous slide of electrode placement. An extended version of that system. 1020 is a very popular system. The advantage of cap is many different montages can be implemented. There are different types of electrodes can be inserted. If you want to use other type of electrode in same slot, you can just change that. Electrodes can be easily replaced in event of failure, right? Because you are wearing a cap, so you can just change the electrode which is not responding, which is not properly, giving the signal. Electrode height can be adjusted and as needed to reduce the impedance. So in a cap, a lot of modification, rather, adaptive usage of electrode is possible. Each electrode must be inserted are removed independently. The wires must be brought together and properly handled to minimize environmental noise or movement artifacts. If you don't do it properly, artifact will be added. So this, I think, yeah, this diagram gives very much clarity. How do we place the EG electrode just at the top, ECOG little inside, just little inside in certain area, and look at the micro electrode. It's very, very deep, inserted, very deeper into the cortex area, cerebral cortex area. So this is how positioning is indicated. And this is what is, cap-like structure is depicted here. Look at here. So you can some electrode is not working in this particular, so you can replace that electrode. So it's up to you. Just connect the electrodes wherever you want to capture or whichever area you want to cover, and accordingly see here. This is how, and then that with the help of cables, you just connect those electrodes and bring the signal with the help of interface, RS-232 serial or whatever, the Bluetooth interface, wireless interface. You just get the signal into the computer for the further analysis. So this is another representation like EEG. You see how much depth actually, which area exactly, what are the areas, where do you insert is the, little more description, further description of what was highlighted in the previous slide. EEG is here on the scalp, and where is actually ECOG is actually, you look at here, this area is a skull. Just inside the scalp is a skull, and there is an area called as dura, whatever. And then there is white color area called as, arachnoid and dura. Between these two, actually, we insert ECOG. And there is a pia, then cortex. This area is cortex. And there is a white matter. and this area is a parenchymal single neuron and local field. The meaning is, if you are going deeper in the area of micro or this particular size where you are getting purely local field potential. So this is just description about how do we place the electrode. EEG signals have generally the amplitude 10 to 20 microvolt, which we get from the scalp directly, and the spatial resolution on the scale of centimeter. It is a very large, and the bandwidth of generally 0 to 70 hertz. More frequencies always in beta and gamma. EEG signals are passed through different fluids, skull and skin. Signals spread and blur because then skin is there, then gel is there, skull is there. So therefore, tissue acts as a low pass filter. The brain tissue attenuating high frequency components of the signal. So this was already, I think, explained in the previous slide. Amplifier in the channel. Brain signals have relatively small amplitudes. The amplitude range is EEG is generally 10 to 20 microvolt. 50 to 100 microvolt is ECOG, and around 1 microvolt for evoked potential. And in BCI application, therefore, all these small amplitudes need to be amplified. Then only we can have a proper interpretation. So I think we have already talked about biosignal amplifier. Sometime amplifier is separated from ADC, and sometime it is combined with digitization also. So the source signal without distortion can be produced, and this biosignal amplifier must have ability to amplify, not only amplify, but also to suppress the noise, because it can also have the filtering capability. It can remove either the low frequency or high frequency component, and it can also. So we can we can use a single electronic conditioning circuit, or we can use separate amplifier, ADC, and filter also, analog to digital converter. So amplifiers have either analog outputs or an integrated output, and biosignal amplifiers are used for neural recordings or instrument. They are called actually instrumentation amplifiers, which are differential amplifiers for measuring a potential difference. So any amplifier which is being used for EEG recording is always a differential amplifier, a kind of instrumentation amplifier. And this particular terminology I was mentioning in my previous slide, that they have a high input impedance, very high input impedance buffer. If you have seen in the one of the previous slide, buffers were used before the amplifier. This is just to avoid the overloading effect in the final differential amplifier. And the differential amplifier that we use must have high CMRR. What is CMRR? Is common mode rejection ratio. What is common mode rejection ratio? Common mode gain divided by differential mode gain divided by common mode gain. So common mode gain must be very less, and differential mode gain must be very high. So the ratio must be very high. A channel consists of, actually, if I can write CMRR for your interest, it is actually written as ADM. I will write slowly with cursor, divided by ACM. Of course, it is expressed in dB. You know about decibel. So this is your common mode gain, and this is different. This is common mode gain, and this one is differential mode gain. So the gain of the amplifier in differential mode signal, and gain of the amplifier when it is amplifying the common mode signal. So this must be, denominator must be very small, and the numerator must be very large. And this is why CMRR is expected as very large quantity. So channel consists of a pair of electrodes. When we talk about channel, because I'm including here that because today we are talking about design. So all those hardware, software are being discussed. So what is channel? It consists of pair of electrodes. Amplifier measures the difference in potential difference to those electrodes. And so the amplifier that amplifies the signal between the electrodes is brought by the channel, which is nothing but, a pair of or a cable. So this is how you look at here. One more interesting diagram. I'm sorry. You see here there are 8 channel selectors. It is bringing from different 8 channels, and then it is all the inputs are going to differential amplifier, then filters, then display. If you want to display here only, it is okay. Or if you want to process something, you can take it to the signal processing unit where you have some memory, where you can have some output unit. Some calibration also can be done so that you can correct the input. You can select a proper channel. You can select proper features here also. And then, of course, the use is always either generating the stimulus so that you can use for certain application for rehabilitation of some kind of thing. So amplifier augments only the different signal and attenuates or eliminates the common signal component. So CMRR is always high, and in dB generally the value of CMRR for the differential amplifier that we use in BCI design is in the range of 60 to 110 dB is generally the range of the common mode. So for each recording, say 128 electrodes on the scalp, an amplifier with 128 channels containing 128 inputs for electrodes, as well as input for reference electrode and an input for ground will be used. Amplifiers with lesser number of channels can also be used. Buffering stage consists of high impedance. I have already explained. That is nothing but a voltage follower. Voltage follower is a common collector amplifier in terms of amplifier that acts as a buffer. Its maximum voltage gain is 1. So those who have studied about voltage follower amplifier, they know that its gain is less than or equal to 1. Ideally, it is 1. So high input impedance that actually avoids overloading. Somewhere it is written, it prevents high resistance source from being loaded down. So overloading effect that can cause difficulty in the performance of the differential amplifier is addressed by using the high impedance buffer amplifier. Amplification stage follows buffering. So when we talk about amplification, it includes buffering, amplification between the two difference. We have seen the circuit diagram. Yeah, same has again come. Look at here. So when we say amplification stage, it includes buffering stage and then amplification stage. And this diagram was already included in the previous slide. So electrodes are typically placed on the scalp with 2.5 centimeter to 10 centimeter between adjacent electrode. So between two adjacent electrode, this may be the spacing, and the ground electrode is placed elsewhere on the head, anywhere. The channel consists of pair of electrodes and amplifier. About the channel, about the spacing 2.5 to 10 centimeter. The bipolar recording amplitude is generally 5 to 20 microvolt, depending on scalp location, inter-electrode distance, and what is brain activity being recorded, EEG. We have already seen the voltage range is the 5 to 20 microvolt. Bipolar recordings are less sensitive to widespread noise. I think this has already come because noise cancellation takes place in the bipolar recording more in a better way as compared to monopolar recording. That cancellation, because of both the electrodes are bringing two data. So if both the electrodes are affected by a kind of noise, so some cancellation of unwanted effect will happen. So the other artifacts are more sensitive to localized brain activity. Monopolar recording, the channels of amplifier use negative input as a common. As a reference, an electrode is often placed on earlobe, which is also called as mastoid. The other electrodes are connected to positive inputs of the amplifier, wherever you want the signal from, the neuronal activity from, there you can connect. So I think this diagram is not new. It is highlighting all different stages of amplifiers, bringing the signals from different electrodes, passing through different filters and ADCs. Is actually the example of a monopolar. You look at here, the reference is 1, and from all individual electrode, one signal is going to the amplifier. So this arrangement is monopolar EEG recording. Typically, the amplitude is up to 50 microvolt and very sensitive to the broadly or wide noise or widespread noise or artifact. They adversely affect the monopolar recording, because if you get two input from here send it to the amplifier, both are likely to be affected by the artifact in the similar manner. So when you actually calculate the difference between the two, that gets cancelled or nullified. So that's the advantage. Filtering, to remove a non-essential filtering, we can also use an anti-aliasing filter. I have already talked about anti-aliasing filter in my previous lecture. Anti-aliasing also a factor when your sampling is poor sampling. This has come in the previous slide when we were talking about signal processing of BCG, ECG, and biomedical, BCI application. So say the frequency is below 100 hertz. If you don't want more, if you don't want less, accordingly filter can be designed, and based on the breakpoint frequency or based on the cutoff frequency, you can pass any frequency either below or after that frequency, depending on what kind of signal you want. So this is little more description about the same thing. Look at here, biosignal is recorded. Electrode artifacts, what are the artifacts? There may be electromagnetic polarization, interference, there may be movement of your eyes, blinks. That may create problem here related to electrode artifacts. Then comes biosignal artifact. EMG, EEG, they have their own artifact. And then electromagnetic interference like power line interference, nearby mobile phone is operating, so that might interfere, etc. So then it goes to high impedance circuit. Again, there may be some white noise, non-linearity, produced by the amplifier. Amplifier might itself can introduce some problem in the circuit. Then it goes to high pass and low pass, depending on what is the requirement. Sometimes when you combine high pass and low pass, this becomes a band pass filter. So many times in EG, you will find we use a notch filter. Notch filter is a filter that is a kind of a band pass filter where the pass band is very narrow. And then it goes to, because when you do this, it can be weakened. So we can again pass. So look at here, we are using a notch filter here. Then if there is any aliasing effect because of all these effects, we can use anti-aliasing, and then we can subject finally to the ADC. And then ADC signal can be given to computer device using any interface, maybe USB interface, Bluetooth interface, that is wireless interface, or as RS-232. You can connect a cable, serial to parallel converter cable RS-232, and you can feed your analog to digital data to the computer. Or you can directly analyze with the analog output in case of the low impedance signal. That is also allowed. So this is one example of how filtering is needed. Look at the first signal, which is is very difficult to analyze. So it's corrupted. So after filtering, we get little better. And after using a smoothing filter, there are some smoothing filters available, which are basically low pass filters. So you see the quality of the signal in the last, at the bottom of the signal. So you want better quality of the signal so that analysis becomes appropriate. Otherwise, analysis could not be proper. And this is why we have to use filtering circuit. Now we are talking about hardware and software. We are talking about interfaces. You all know there are, if we talk about, protocols, there are IEEE protocols, which are actually rules that govern the exchange of information, because the interface is providing, connectivity between your BCI and the outside world, computing world. So there must be some protocol set up rules. Computer serial ports like US RS-232 for serial binary data transmission. This is very robust and easy to use, but the difficulty with RS-232 is low data transmission rate that limits the performance of the system, and therefore it can be limited to only few channels. So the data rate, because of low data transmission, the data rate is very slow in case of RS-232. Then comes Bluetooth. It's a wireless protocol transmitting data up to 100 meter. The biggest benefit of Bluetooth is the mobility is always involved. You don't need wire, so you can always operate in wireless mode. So mobility is a big benefit for biosignal recording because subjects can move freely with the recording device. However, such wireless and mobile systems entail high power consumption. Whenever you all understand, whenever your device is on wireless, it requires always more power consumption, more power. So the bandwidth, so the power consumption will be more in case of wireless. Bandwidth is also distributed among, say, what I'm saying is with the wireless, you know it very well. You can connect n number of Bluetooth devices. Suppose you have a Wi-Fi in your home, you can connect 10 laptops, you can connect 10 number of wireless devices. So in that case, what happens, the entire bandwidth, your data rate, Mbps or Gbps, is divided. So that is a constant maximum data rate will be divided among all those Bluetooth devices. And USB is a serial bus standard. USB, you know that all of us use the USB. USB 2.0 is a standard that provides higher bandwidth than Bluetooth or RS-232, and therefore it allows more data to be transferred with the higher sampling rate, and even it supports more number of channels in the USB interface. And then you need to have a client hardware. When you are having interface, you need to have a client hardware. What will be your client hardware? User hardware. That will be nothing but computer or a microcontroller. The client hardware can perform a number of tasks, like it controls data acquisition, it stores the data from ADC, it processes the data to extract the features, and then finally it has to translate them into output commands, and also sending those commands to the application device. Say the application device is your, what is called as a wheelchair or some other arm that needs to be controlled. So all these are functionalities of the client hardware that does it. So then we need to use software, and software is nothing but the mathematical model or the programming or the mathematics that will be used to analyze the signal, and after analysis of the signal, we'll produce the real-time output. Why do we want real-time output? That actually controls the operation or the intended application that the user wants with the help of BCI. So what are the functionalities for which we use the software? We use software for data acquisition that includes amplification, digitizing, transmission, and storing of signal. We can do the signal analysis that includes extraction of features, analysis of features, converting into feature vectors, translating those features, classifying those features into suitable commands, then generating the output, maybe either movement of a cursor or movement of a controller or movement of a wheelchair, or also can provide a feedback signal. So all these are done by the software part. So you need to have a suitable software inside your, that client hardware has, to be embedded with the software. And then of course, operating protocol that is a set of rules that helps to configure and parameterize the acquisition, entire analysis, output components, that also determines onset, offset, and timing operation of the system. And practical aim is to operate all the devices to provide communication, that is very good communication or control capacities to the people, even with the severe disabilities. That's the application. So I think we have come to, and then of course, assistive technology is a very interesting application where we use the BCI there. You see here, active BCI is used for replacement and restoration, and passive BCI is generally used to enhance certain ability which is lost. And all these particular portions I have already covered. So I think the next part, which is related to ethical issues, we can cover in the next part, where I'm going to talk on a practical implementation of cognitive workload assessment, that is work using EEG signal analysis and by using MLAI model and optimization methods. So we have come to